sorry. And this is chapter 6. Okay, so chapter 6 is about DNA and gene expression. So first of all, let me explain to you the concept of gene expression. Okay, so your DNA, right, is a bunch of information, right? And one more time, I hope you remember. Gene is a part of DNA, and one gene is a code for which molecule? We talked about that. One gene corresponds to one.
Oh, that's easy.
Yes, one gene is one what?
Me or in general?
You can answer, David. I will be happy if you answer that question.
Oh no, it's
I
like
got it.
yeah,
Yes,
it's
please.
too easy. I want to let someone else answer it.
One gene is one protein.
Excellent, Catherine. You made me so happy this morning. Yes, one gene is one protein, right? But the thing is that once the very first cell starts to divide, right? And there are more and more cells. And each cell acquires different shape and different functions, right? So not all genes are expressed in every single cell. What makes cells different is different expression of the genes. Expression means the gene is active, right? Gene is actively coding for protein. And the protein that are coded, for example, in red blood cells are not the same proteins that coded in neurons, and not the same proteins coded in skin cells, and so and so and so and so. Plus, your own DNA, like total DNA, has a bunch of bulk DNA that is never expressed. It means like just dragged from generation to generation without actually showing up, right? And again, in one generation, genes can be silent. Another generation, genes can be expressed. Okay, so that's what also makes the difference, you know, between family members, right? The expression of the genes. So one more time, expressed gene is an active gene. And this is the core, right? The DNA molecule. So DNA has information about the whole entire organism, and every single cell in the organism have the same DNA. Again, it's just which genes are expressed, what makes a difference between them. So these two guys shared Nobel Prize. Yes. They shared Nobel Prize a long time ago, right, for actually coming up with a DNA structure, with a model of the DNA, which was a huge breakthrough back then. And in these days, you can't even like imagine how to live without having information about genetic material. Okay, so DNA stands for desoxyribonucleic acid. And it's made of sugar, which is a pentose, which means 5 carbons in the sugar. And in the case of DNA, it's desoxyribose. There is a phosphate group, and there is a nitrogen base, or what they call, like, the whole thing is called nucleotide. The whole entire thing is nucleotide, which is made of nitrogen, contain base, pentose, and phosphate group. And there are 4 bases, right? Nitrogen bases or nucleotides: adenine, thymine, guanine, and cytosine. And DNA has 2 strands, right? And 2 strands literally like wind around each other, and they held together by hydrogen bonds. Hydrogen bonds are formed between adenine and thymine, guanine and cytosine. So that's called a complementary rule, which means that adenine always goes with thymine and guanine always goes with cytosine, again, for the reason of the hydrogen bonds. And that's what we just started the classes, right? So genes, section of DNA that have instructions for making proteins, right? Human proteins, onion proteins, fruit fly, Drosophila, amoeba, salamander, right? The only difference between these organisms is the DNA, right? And the proteins that this DNA codes for. And the whole collection of genetic material is called the genome, right? So entire DNA. Genome could be broken down to chromosomes. Chromosomes are units of DNA and proteins. In gene is a piece of DNA, region of DNA, or like in genes, you can say they sit on chromosomes. And where they sit is called the locus, right? So this is the whole entire genome. This is part of genome, chromosome. This is the region of chromosome, or DNA, it's a gene. And this is the protein that this gene codes for. And let me explain something. Let me get to whiteboard. Okay, so and let's talk about humans. And again, even though it's like biology is much more general than human, it's about all living organisms, but we're gonna talk about humans, okay? So we humans have how many chromosomes, guys? You can tell me. Guys?
We have
Wait,
23?
say that. I know that answer.
Again?
Chromosomes?
How many?
Break the fuck.
How many?
23?
46?
36.
46. Yes, we have 46 or 23 pairs, right? And out of this 46, 44 are called autosomes. Why autosomes? Because they are not involved in determining gender, okay? So they code for a bunch of things, but they don't really determine whether you're male or female. And there are two sex chromosomes. And sex chromosomes for female sex chromosomes are XX, right? And for males, it's XY, right? So basically you can say if you're a female, you are 44XX, and if you're male, you are 44XY, okay? Now, let me show you something. So,
Wait, I'm sorry, Professor. Can you go back anyway?
I should, but like, I mean, I'm gonna continue with the same thing. So,
Okay.
so let's say mother, right? So mother, let's say she is female, so she's 44X1, X2, right? In her egg cell, she can contribute only half of her genetic material, right? So in the egg cell, how many chromosomes will be? How many chromosomes will be in the egg cell? Tell me.
Is it 26?
It's gonna be 22, right? And one of the X's, basically total will be 23, right? So half.
Okay.
So it can be either 22 and X1 or 22 and X2, right? She doesn't know which one, like, will go. So father is 44XY, right? So in the sperm cell will be 23X or 23Y, right? Okay. So if this one will meet with this one or that one, it's gonna be 44X1X or 44X2X, right? And if this one will meet this one or that one, it will be 44 x1y or 44 x2y, right? So this two will be who? This will be males or females?
It'll be a female.
Yeah, this will be girls, right? And this will be
Male.
boys, right? So, which means that who defines the gender of the child? Mother or father?
Father.
Absolutely, right? Mother gives only X chromosome, right? It's father who gives either X or Y. Can father control that?
No.
No, no,
No?
really. Right. No way father can control it. I mean, there are techniques. If let's say in the family there are 10 boys and they want a girl, right? So they can turn to some institutions that can help with that, but no, it's not under man's control, right? And another thing what you can see, so chances to have boy or girl, what are they?
50-50?
Yeah, ideally 50-50, yes. But do you think, is this the case?
Not all.
Does it change with
No.
the order of
What?
what you had?
Okay, I'll put this way. So what do you think? Let's say for 100 newborn girls, there are more newborn boys, more or less?
Probably less.
Actually more.
Oh.
More.
Statistically, more boys are born than girls. And I can explain actually why. Catherine, can I erase this?
Yep.
Okay. So let me explain you why there are more boys born than girls. It's not also like people want, for whatever reason, like especially fathers, they like to have boys in the families. Okay, but nothing to do with biology. So Excel, do you remember I told you that you inherit from the mother all their cellular contents? Remember that? Remember when we talked about mitochondria and mitochondrial DNA? Do you remember that? Okay, so Excel is huge. It's also it's the largest human cell, right? And it's actually the only visible cell. While sperm cells are really tiny. So why are they so tiny? Because they don't carry any organelles, right? They don't carry cytoplasm. What do they carry? They carry 23 chromosomes, tightly packed. Now, if you look at the X chromosome, it's actually a very big chromosome. Y chromosome, which defines the gender, is tiny. It has only 50 genes. It's a really small chromosome. So if this one carries X and this one carries Y, which one is heavier?
Can you repeat, Professor?
Okay, I told you that chromosome X is huge. It's a big chromosome. It carries a bunch of genes. Y chromosome is a small chromosome. It carries only 50 genes only. So if the sperm cell carries X and that sperm carries Y, which one swims faster?
X.
Okay, one more time. Which one is heavy? Which one is heavy? The one that carries X or the one that carries Y?
The one that says X.
Exactly. This one is heavier, right?
And
So,
why be faster then?
of course, Y will swim faster, right? So if Y can swim faster, it has more chances to hit the X, right? So that's why statistically there are more boys born than girls.
That makes sense.
Okay, but the thing is that I should say by age like 50, they're pretty much the same, like the ratio is the same. And by age, let's say 80, 90, there are definitely more females than males, right? So kind of like, I think the nature tries to compensate this thing, right? Okay, any questions here, guys? Any questions? Okay, so let's continue. Actually, sorry, you know what? I'm sorry, I want to go back to this one. Okay, so when you talk about genetics, so genetics is a science of genes and inheritance, right? And inheritance is actually, inheritance is way of transmitting genetic information. From parents to offspring. So parents give genetic material in form of chromosomes, right? So chromosomes, they carry genes. So gene, in the sense, is unit of heredity. Now, let's say, again, pretend we look at the first two chromosomes. Okay, so let's say this is pair number 1. Remember, you have 23 pairs. This is the very, very first pair, right? Now, try to understand that in each pair, let's say pair number 1, pair number 2, pair number 3, and so on, so on, so until like 6 chromosomes. This chromosome is called homologous. Homologous chromosomes, it's a pair that carry the same genes. So every single gene is duplicated, right? So let's say you have a gene for your hair color, right? So you have a gene for hair color from the mother, and you have exactly on the same spot on the paternal chromosome, you have a gene that defines your hair color, right? So having pair of chromosomes means you have pair for every single gene, okay? So can I go to, Catherine, can I go to,
Yeah, okay.
okay, so in this sense, what you can see here, right? So first of all, we have something called characteristics, right? Characteristics is something like your height, the way your body is built, your eye color, your hair color, the shape of your ears, the shape of your nails, right? Trait is a version of characteristic, right? So trait can be, let's say, round nails, oval nails, curly hair, straight hair. So these are traits, right? And that's what you pretty much inherit from your parents, right? You inherit different traits. Or you can say you inherit different versions of the gene. So different version of the gene that codes for the same trait is called allele, right? So allele, let's say this one in flower codes for orange petals. Allele 2 codes for yellow petals. Allele 3 codes for purple petals, right? So you can have, let's say, again, depends on what you inherited. And I should say it's always like, it's like a dice, right? It's every time it's a new combination. I remember my cousin, who I didn't see for a very long time since childhood, right? Because she lives in Cyprus, we live here. So she came to visit with 3 daughters and from the same father. So the first one, the older one, has really dark hair. The second one has like, let's say like brownish hair. And the third one is blonde, right? So the same family, right? But somehow the combination, genetic combination, basically came up with different versions, right? But if I go back to what I was talking about, right? So homologous chromosomes, right? And this is side of allele from the mother. This is allele from the father. Let's say this is like ears like this, and this is ears like that, right? So different versions, different alleles. Is it clear, guys? Did you get the concept?
You said different versions, different what?
A different version of the gene is called allele. Allele, let's say allele for round ears, allele for pointy ears. Allele for oval nails, allele for round nails, and so and so and so. Again, you never know exactly which allele you received from your parent, right? I think next week we're going to talk more about how genes interact, but at this point I just want you to understand that what you inherit genes. Genes code for proteins, which sometimes corresponds to trait, right? Trait is, again, variations in your looks, variations in your physiology. And these traits, if you look at the level of the DNA, they basically called allele, which is different version of the gene, right? Look about the eyes. And eyes might be not the best example because our eye color is determined by more than 100 genes, right? But let's say trait, I could be blue, I could be brown, I could be gray, right? Or you can say allele for brown, allele for blue, allele for gray. Yeah, guys, did you get it?
Mhm.
Okay, now, well, you can also see that not all DNA contains instructions for proteins, right? And this one, you see, genome size, right? So fruit fly has, let's say, 180 genes. It's something like weird. Genome size. No, human genome size is definitely more than, I don't know what they compare, but basically you see amoeba has a bunch of genes that it doesn't really use it. That's which means I told you there's like bulk DNA that you just carry from generation to generation, right? So this is percentage of coding DNA. So human DNA is only 2% on that codes. Fruit flies have actually more, roundworms even more. This is some plant. And E. coli, actually 90%. But E. coli makes sense, it's only like one cell. So it's gonna use all its genes, right? So basically I can say 98% of our genes, we don't really use much. This is crazy. Okay, so what is non-coding DNA? Non-coding DNA is we carry something like a bunch of viruses. And there's something I want to explain to you about viruses at this point. we're gonna talk about, we'll talk about a little bit more later on. Okay. So, virus. Virus we consider is like infection that causes diseases, right? But what's interesting about virus that viruses are not really considered living organisms. So it is not a living organism. What is virus? Virus is
It's something that is just designed to replicate all the time and then jump to another host.
yeah, yes, a genetic material enclosed in protein shell. So virus, it's parasite. What does it mean, parasite?
It needs a host.
Exactly, right? It's like, you see, parasite is something that kind of benefits on your behalf without giving anything back, right? So it's a parasite because what viruses do is they use hosts for their own benefits. And before we basically get to host, I just want to explain to you the viruses. It's protein shell, and inside could be piece of our DNA, or virus can be an RNA virus, right? It's either DNA or RNA virus. And viruses are very specific in terms of finding the host. Some viruses infect only plants. Some viruses infect bacteria. Some viruses infect animals, specific animals. For example, pets can carry viruses that actually can infect humans, right? And human viruses can infect pets viruses. Sorry, they can infect pets. So how do we pick up viruses? There's something called mode of transmission. Okay, so if we talk about human viruses, how can you catch a virus?
You catch a virus through being infected from another host.
No, how? I mean,
Well, usually, here's an example from the coronavirus. The coronavirus transmits itself through the air. It could also transmits itself through the eyes, through spitting, sneezing, genetic material getting through, and then you eventually get sick.
no, it means like airborne.
Yes.
Airborne,
Sorry, I didn't have the
or
exact terminology.
direct contact.
Sorry, I didn't have the exact terminology. I'm sorry.
Okay,
about
so let's say if I ask you, for example, sure you heard about, I'm sure you heard about hepatitis virus, right? The one that
Yes.
infects liver. What's the mode of transmission for that one?
Needles.
Needles, yes. What else?
Sex.
Sexually transmitted. And also depends which one, like hepatitis B and C, sexually transmitted, right? Or through the blood. Or virus could be transmitted, what we call oral fecal route. That's, by the way, also route of transmission for poliovirus. Thanks God we don't have much polio anymore. They're much more common among kids because kids, like, the hygiene is a bit off, right? But there's something, let's say, kid goes to the bathroom, doesn't wash the hands, right? Touches the face of another kid, and here you go. That's how polio spread among the kids much more than among adults, because adults used to wash their hands, right? Bloodborne. It could be sexually transmitted. Okay. And once, let's say, you pick up virus, what happens? It also matters how many viral particles you have to pick up to get sick, right? So sometimes like few viral particles won't do much, but you have to be exposed to much more. So once you pick it up, viruses don't know. They don't know where to go. So if it's airborne, you just inhale, right? And they get down to your airways. If this is with the food, it eventually gets into your blood and disseminated around the body. But anyways, virus has to find the right cell. And again, they're quite specific. So it finds the cell, and cells don't want viruses. They don't like them, right? So they have to sneak in. And they have to learn which trick to play to sneak inside the cell. If you remember receptor-mediated endocytosis, that's how HIV virus sneaks into the T cells, through receptor-mediated endocytosis, pretending to be, let's say, cholesterol molecule. So they sneak in the cell, and it's what matters. There are basically two types of viruses. There are active, and there are latent. What is active virus? Active virus gets inside a cell, releases its genetic material, right? Uses the cell to duplicate this genetic material. The cell does it for the virus. It turns like a slave. And as a slave, this cell gonna make new viral particles. So one virus enters, 1,000 viruses leave the cell. And when they leave the cell, they usually kill it, okay? So I told you, your cells don't like it. And immune system will mount an attack. Once the immune system mounts an attack, that's when you get sick. You experience all the symptoms like fever, coughing, sneezing, feeling miserable. It's not about the virus. Virus just kills the cells. But the immune system, like, initiates a war, right? And sometimes this war can make you quite sick, right? Can sometimes make you can make things even worse than it should be, okay? So this is the active one. And I should say that RNA viruses, they're usually those that are active. Okay? And RNA viruses are those that are not stable. They tend to mutate and to change. Like viruses that cause like flu, they're RNA viruses. So each year you have like a new addition, right? What about latent? Latent, this is usually RNA. That doesn't work. This is active. What latent virus will do? Latent virus will sneak in, right? And without doing much, will go into the nucleus. In the nucleus, you have your genes, right? It will just insert itself into your genetic material, right? Whenever the cell divides, this virus will divide and divide and divide. So more and more cells, after divisions, will carry this virus. And then virus one day can wake up. And once it wake up, it can cause trouble, right? This is an example of HIV virus. Okay, HIV virus sometimes takes years to develop the disease, right? But I think a very good example is herpes virus. Herpes virus is a big family of viruses. We usually know the type 1 and type 2. Type 1, we call it above the belt, and type 2 is below the belt, right? If I talk about type 1, how people usually get it? The one
Oral?
that's oral, yes, the one that causes, yes.
Sores, open sores, right?
Yes, this is type 1.
Professor, look at this. Oh my God.
What?
I'm so studious. You see that?
What is that?
That's all bio homework. I'm like,
What? Okay.
my fault. Yeah, that's pretty much it.
Okay, so how do you get type 1?
Skin to skin contact, right? With the herpes.
Correct? Yes. Skin
Where's
to
that
skin. I
music
mean,
coming from?
I should say, and trust me, guys, type 1, I think everyone has it. Okay. Everyone has type 1. It's so easy, right?
Type 1
Person
what?
who has sore, right?
Apparently, yeah, that's a
Person has
herpes.
an outbreak, yes. So first of all, you can get it only from the person who has an outbreak, right? So the person who has outbreak shakes your hand, right? And you, like, without thinking much, touch. And people tend to touch their faces all the time, right? You touch your face like this.
So, Professor, we all got herpes.
We all have herpes.
You got herpes?
Me? Type 1, I think so. As I said, almost everyone has it. No,
I heard Ingrid has herpes.
that's
Excuse me? What?
We all have it. What are you talking about? I have herpes too.
You're bugging out.
Those who have it, only half will ever have cold sores.
Ah,
All of the virus never experienced because virus is dormant.
right.
The virus can sleep forever.
Oh.
Virus can wake up from time to time.
Professor, can I ask you a question? I was talking to my advisor. My advisor said that you can't contract herpes if the other person's symptoms doesn't show. I don't know how true that is.
That's absolutely true. You can get only when the person has an outbreak.
Oh, really? That's crazy. Okay.
So
Thank
before
you.
you tell someone, just take a look.
I'm gonna just tell them, listen, are your herpes dormant? And if they say yeah, okay. But if they say it's not, then respectfully, I cannot.
Yeah, you can ask for a note from the doctor as well.
That's true. That's true.
Okay, so what I'm trying to say here is that you understand what viruses do, they just insert their genetic material. And people from, I think from the dawn of evolution, carry these dormant viruses in their DNA, right? So this is our DNA inside the nucleus. So very large percentage of this DNA are the viruses that we accumulated. And some viruses accumulated for bad, some viruses accumulated for good, actually. Some viruses that humans picked up actually provide resistance to certain infections, right? So this is non-coding DNA, and I said most of it, large amount, are pretty much viral DNA. Okay, so there are two important definitions that, Ingrid, what is that? Okay, so genotype. So genotype is all genes that we carry, right?
Mm-hmm.
Now, phenotype is physical manifestation. Physical manifestation pretty much reflects your genes, right? I think a good example will be with dogs, right? So dogs are the same species and the DNA is related. But for example, Great Danes, they have one phenotype and Yorkies have another phenotype and Poodles have another phenotype, right? So it's usually quite specific to species, right? The phenotype that you present, but it's one more time reflection of your genes. Okay, and this is pretty much like the whole story of today's class. So I'll summarize it for you, and then we'll take a look, step by step. So from gene to protein. So from gene to protein, it's actually sometimes called the central dogma in molecular biology. So from gene to protein. And remember, one gene codes for one protein. Now this is a cell, right? So in the cell, there's a huge nucleus with a DNA. And then around the nucleus, you have endoplasmic reticulum, right? And on the endoplasmic reticulum, you have protein making machines. How they called? Guys, how these protein making machines are called? Hello, are you there?
Sorry, Professor, it's a Tuesday. We're not used to this.
You had a
Can you
quiz.
repeat, can you repeat your question, please?
How protein making machines are called? Those that sit on endoplasmic reticulum.
I'm sorry, I was on mute. Is it ribosomes?
Wow, David. Wow. Respect. Yes,
I got it right? Of course, you know.
they called ribosomes. So ribosomes, they build proteins, right? Now, for ribosomes, what they need instructions. They need instructions how to make these proteins. But genes, right? Genes are inside the nucleus. So it basically happens in order to make a protein, the DNA has to, first of all, has to get activated, right? And the first step in protein synthesis is called transcription. The transcription gene kind of like wakes up, and then DNA gene is copied into RNA molecule. So it's like, you know, I think it's good to compare it to, you have in the library, right? You have books. And let's say you find a book that has a bunch of recipes. And you look for the recipe, let's say how to make a stew. You don't take the whole book with you out of the library. No, you just go to the copy machine and you make a copy, right? So to deliver instructions to the ribosomes, you don't have to pull all DNA out of the nucleus. What you do, you make a copy of the DNA, right? And once this copy is made, it leaves the nucleus, right? It leaves the nucleus. Remember, there are nuclear pores, the openings. It gets outside. The first ribosome that sees this will just kind of like snap, you know, catch it. And then this ribosome will follow the instructions and build a protein. This is called translation. The translation is ribosome builds protein using RNA code. Okay, so the whole story is, again, DNA is inside the nucleus. Ribosomes are outside. Ribosomes need the instructions. The instructions come in the molecule of RNA. In this case, this RNA is called mRNA. mRNA is a special RNA, which is a copy of DNA. M stands for messenger. Okay. Messenger RNA that is literally a copy of one gene. Okay. RNA is a very useful molecule because RNA pretty much reflects which genes are activated inside the cell. Because DNA is said it's a bulk. It's like whatever is there, a bunch of things. RNA is one specific molecule. Okay. Can I go to the PowerPoint, guys?
Yes, go ahead.
Catherine, can I?
Yes.
Okay. This is like what I was talking about. Grandmother cookbook, right? So all genome. Then there's an enzyme called RNA polymerase. This enzyme does transcription, copies one gene, right? And you see DNA unwinds, opens up. And from one strand of DNA, RNA molecule is copied, right? This RNA kind of like snakes out of the nucleus. Once it's outside the nucleus, ribosome will pick it up. And this is translation, right? So from RNA to protein. Is it clear?
Yes.
Yes, guys, is it clear? This is again, this is the whole point of this particular presentation, right? So transcription. So transcription, the first step in transcription is to recognize which gene has to be activated and has to be copied. So each gene has a beginning and has an end. Beginning is like capital letter in the sentence, right? The beginning of the sentence. And end is like a period, right? So RNA polymerase binds to the beginning. It's called promoter site. RNA polymerase is a powerful enzyme that sits inside the nucleus. Then it transcribes. Again, RNA polymerase basically builds a new molecule, which is a copy of DNA. Then it stops, right? So from the beginning to the end, so it stops at their termination stop, right? And so you see, the last step is an RNA. It's extra processing, okay? Gets extra processing. And this extra processing is actually quite often DNA has to be edited. It has to be altered. Okay, guys, it's 9:33. Let's say 10 minutes break, 9:34 till 9:45, okay? And listen to me. Even if I put myself on mute and turn on the camera, it doesn't mean that my laptop is not next to me and I don't hear what you're saying.
Yes, David.
But we're on a break, so it
Submitted the
doesn't count.
homework. I
Right.
submitted at least half of the homework. I couldn't really do the other half because it was
Love, can we talk about this later? I
okay.
promise. Yeah,
All right.
my special time and we'll talk about it.
All right.
Professor,
Yes.
I'm good. Attention on the timer. Under the timer. 2 more days.
What is going on?
wait, that's his mic or that's my phone?
No, that's his microphone.
Crazy. No, that's definitely his phone. I was bugging too.
No way. That's my phone, yo. Yeah, yeah.
Yes,
Nah,
yes.
it should
Up to
be.
50. I'm
It
sorry.
sounded like this.
Now you know
the
the smoking commercials where old people
dude.
talking like that. That's what you sound like.
Is their camera off? Does it count? Because we're on break.
Bro, it still counts.
Professor, you gonna take off points if we curse?
Some mood said whoever's mic that's from is from Area 51. That's pretty funny.
All right, guys. Do you like cookies?
Lab, do you have herpes?
I do not have herpes. I've never had sex, sadly.
Oh no!
Imagine.
It's dormant. It's dormant. You never know. It's just sleeping. The beast within you is sleeping.
If I do have herpes, I'll take a load of drugs.
Oh no!
Oh, nah.
The only thing I have ever had in bed is this thing. But we don't do any shit. All I do is just do that.
That's Charmander, right?
No, it's Piplup, goddammit.
Bro, what?
That's not Charmander. Oh, my
Did
fault,
you just
bro.
say
Nah, that's
Charmander?
My fault. My fault.
I'm a Piplup!
Yeah, I love this class. This is the only Zoom class I like. Yo, Lab, you want to hear some Spanish song? Damn, that's crazy. Just left me unheard.
I do want, the best carne asada I've ever had
Yo.
is
Yo, that's
the
mad
best
racist.
the best carne asada. Yes, it is. Sadly, I'm not racist, but I have a tendency to say very stupid racist things. Like,
Don't worry, don't worry. I give you permission.
But the best carne asada I've ever had is probably near the campus.
Near the campus at BMCC? That's crazy. That's definitely not the best carne asada you've ever had in your life.
Well, of course not. But you got to get your fix.
Definitely not in Manhattan. Definitely not in Tribeca.
Yeah,
That's
Tribeca
white
has
people meat.
horrible
Go to
food.
Queens for that.
Yeah, you got to go to Queens for that. You definitely got to go to Queens for that.
Now,
You did
I
not
did
just
have
say
good
that's white
Chilean
people
food in Queens or maybe Brooklyn. I forgot. It's called La Rojas, La Todos Rojas. I can't remember. But man, Chilean bakeries are
Oh,
the best.
I know what you're talking about. You're talking about Doha or Sabor Latino.
Yeah,
You been
that's
there?
no. That's the. I made friends with this great Chilean guy, and he's the only friend I've ever had that was genuine in New York City. He would take me to the places, and my parents were like, "Where's he taking you, man? Where's he taking you?" It's like, "Why do you have to go out like this? Why are you going out to a Latino place?" Oh, it's like,
Dad, your parents racist like that? That's crazy. Let me speak to them.
it is. It is very sad as to how racist a lot of well-off people are.
So it's true, right?
Self-made people. It's like, oh, we don't want. No, you shouldn't hang out with those people because they're idiots or some shit like that. But I am nearly done with BMCC. Nearly done. Nearly done. Nearly
Damn,
done.
You just called us idiots? That's crazy.
No, I'm not calling you idiots. I'm nearly done with BMCC and I don't want to be here anymore.
No,
What am I gonna do?
I felt offended, and for that, I'm going to leave.
Okay. Me and my big fuck.
How RNA can provide this function. And oops, just 1 second, guys. I'm sorry. I think I left my pen. Give me 1 second. I'll be right back.
Yo, can y'all still see me like this? Because I'm not on the app. I'm just asking. I bet.
Okay. So now this is a little bit not straightforward. So I explained to you that there is an mRNA, messenger RNA, which is basically the DNA copy. There is also something called rRNA. rRNA is ribosomal RNA. RNA that ribosomes have. And there's a tRNA. tRNA is transfer RNA. So let's say this is the RNA molecule, and this is a ribosome. And RNA molecule is a copy of DNA, so it can be ATP, C, G, G, C, A, C, T, G, A, C. Okay. How ribosomes basically do this whole thing. And there's something interesting about that. Every 3 letters of RNA correspond to 1 amino acid. This is called codon. Codon is 3 bases. Is 1 amino. So ribosomes on their own can't do much. What ribosomes can do, they can combine, they can connect 1 amino acid to another. But these amino acids are delivered to ribosomes by tRNA. So tRNA is a special transfer RNA. It's an RNA that has 3 letters, let's say ATT, and these 3 letters carry amino acid. So transfer RNA can pick up amino acid based on its own code. If this code matches the code of the RNA molecule on the ribosome, this amino acid will be included. So if I go back to PowerPoint, what you can see, this is messenger RNA, this is transfer RNA. So remember, they're complementary, right? So A goes with T, but in case of RNA, instead of T, there is U or cell. So they have to match 3 letters on RNA, messenger RNA, 3 letters on transfer RNA. Once they match, amino acid will be picked up by the ribosome, right? And then messenger RNA will shift the next code, next amino acid, next code, next amino acid. I'm gonna show you a video. This is like, there is really something mind-blowing video on that. So you see, there are 20 amino acids. Each amino acid has a name. You don't have to know the names, don't worry about the names. So 3 letters correspond to 1 amino acid, and usually there are more than 1 code. Each amino acid might have, let's see, serine has 4 codes. Phenylalanine has only 2 codes. Leucine, 2 codes. Proline has 4 codes, okay? So, but that's the whole idea. From letters, from like nucleotides on DNA, then RNA, each 3 letters mean 1 amino acid. And depending on the gene, right, the protein might have 100 amino acids. It might have 1,000 amino acids. It really matters what gene it codes. So right now I'm gonna show the video. I'll share the sound with you. And I hope it's gonna make things more clear. And again, this is something that happens. It happens in your cells all the time. And this video is a real-time video. So something that really happens in real time.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules. What you are about to see is DNA's most extraordinary secret, how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Okay, so did like make more clear what I was talking about this video? Yeah, yeah. So don't really worry again about all the details. Just two important concepts. Transcription happens in the nucleus. DNA is copied to RNA. Translation happens outside the nucleus, right? Where ribosomes pick up RNA and translate the code to amino acid. So this translation is possible because of the transfer RNAs. Each transfer RNA can carry one amino acid, right? If code on transfer RNA matches code on messenger RNA, this amino acid will be added to the protein chain, right? To the amino acid chain. Any questions here, guys?
No.
No.
Okay, so this is basically the diagram again. Beginning,
So
yes.
the RNA structure is or the mRNA is basically attaching itself to the RNA.
So what you can see here, like in this diagram, look, this, for example, here, this is the ribosome, right? And that's the beginning of the chain. This is usually the first amino acid in the chain is methylalanine. So this is the first one. So this chain is already building. So for example, this one is done, it's gonna leave, right? This one, you see the code matches on the transfer RNA, matches code on the messenger RNA, add it, right? The next one in the row, if code matches, add it. The next one comes, add it. My question, how they know, like how transfer RNAs know that they have to get to the ribosomes? I think there's still no answer. Like how the whole thing is so tightly orchestrated, right? This is an amazing thing. And what you watched, I hope again you paid attention, was real time. It's something that is constantly happens in your cells, right? Isn't it mind blowing?
I'll try to understand it better.
So, look, one more time. A ribosome is basically, they're not specific to any proteins. Ribosome can make any protein, just feed it the RNA. RNA has the code, right? Three letters, one amino acid, right? So to build a protein, they need supply of amino acids. Each amino acid is carried by transfer RNA, right? Each transfer RNA has amino acid and the code for amino acid, another 3 letters. If these 3 letters match 3 letters of messenger RNA, amino acid will be added, right? Another transfer RNA code matches, amino acid added. Another transfer RNA, and so until it's done. As I said, big mystery, how transfer RNAs know that they have to get to the ribosomes. Okay, and so there are a few more things, and then I'm gonna talk about biotechnology, but as I said, I'm not gonna cover the whole chapter. So gene expression. So gene expression, as I told you, is when the gene is active, which means it's actively transcribed and translated, right? Gene regulation is actually a way to turn the gene on and off. Usually there is a special molecule added to the beginning of the gene. So once this molecule is added, the gene is silent. When this molecule is removed, the gene becomes active again, okay? And sometimes certain life events, environment can actually affect this gene regulation, turning your genes on and off. MicroRNAs is something from molecular biology.
Wait, Professor, I'm so sorry. Can you go back for a few? For like 5 seconds? Yeah, gene regulation. So gene regulation falls under nature versus nurture. That's all right, that's pretty cool. All right, that's all I wanted to know.
Yeah, yeah,
All
it
right.
is. Like, I mean, as you're gonna see, like in a few seconds, I'm gonna get to like alteration of genes, but you see, genetic information that is, let's say, carried in your reproductive cells, right, in sperm cells and in egg cells, this information could be altered by your personal experience. And that was proven. Basically, let's say a woman who suffered, I don't know, she suffered some serious stress, genetic material in her egg cells can be affected by the stress and will affect, let's say, her children. Yes. Okay, so controlling gene expression. So there is something called transcription factors. Transcription factors can like affect their expression of the genes. There's a positive control factors which initiate and speed up gene expression. And there is a negative control which block gene expression. As I said, certain molecules can bind and control and basically cause a gene regulation up and down. And operon structure is actually a C promoter. This is the beginning of the gene. This is where the RNA polymerase binds to make a copy. Operator, that repressor protein can bind and block, for example, gene from expression. And then the genes, this is the region that is copied into RNA. And as I said, if operator has any proteins there, they can either speed up or block the gene expression. And that's what it basically shows. You see operon on, and this is, for example, lactose, right? Lactose is a sugar from their milk, right? And this, let's say, gene needed for lactose metabolism, right? And lactose is not present, it blocks, right? Lactose present, operon on, right? So it's not really, doesn't really matter what happens here, but I'm just saying that this region of the gene, that's where it can be regulated. And this is pretty much the same thing. Now here, mutation. So mutation is change, it's alternation in the DNA sequence, right? And any alternation in the DNA sequence can actually cause faulty protein, right? Altered protein. Remember, you actually do anything, any manipulation with their nucleotides, right? And that's gonna code for wrong RNA and from wrong RNA to wrong protein. Mutations usually not really beneficial. It depends on the mutation, but some mutations could be quite neutral, don't do much. But mutations, change in the genes, also considered driving force in evolution, but it takes usually a long time to see their effects. So you see, mutation in the gene can change the, since fruit flies don't have that many, I think they have only 5 chromosomes, right? This mutation basically affects their shape of the eye, right? Which can cause problems in the vision, right? Mutations can happen in regular cells, somatic cells, so non-lung cells. And mutation that can happen in any random cell that is not sex cell, usually confined within the cells. If mutations happen in sex cells, they can affect the offspring, right? So something that you give to next generation. And mutations can be point mutation. Point mutation is on the level of the, and mutations can be a much larger scale. on the level of the chromosomes. And on the level of the chromosomes, these mutations called chromosomal aberrations. So this is normal DNA, right? Normal DNA, normal code, normal protein. One mutation on the level of the DNA could be substitution. You just get the wrong nucleotide, right? Wrong nucleotide, you get wrong amino acid, which can affect the function of the protein. Insertion, you have extra nucleotide, and that's going to screw big part of the protein. The same happens when there's a deletion, removal, okay? But as I said, mutations happen all the time because your cells divide, DNA is duplicated, and this is actually like an ongoing process. We have a whole mechanism in our cells that makes sure that DNA is copied correctly, but still, there's nothing 100% mistake proof. Chromosomal aberration is like something that breaks number of genes on the chromosome, right? If one gene is deleted, or gene is relocated, or gene is duplicated, that has like a greater impact on entire function, not only one protein, but like a bunch of things. And what can cause mutations? Mutations, you see, spontaneous is just a mistake that happened during DNA replication. But there are a bunch of mutations that can be caused by environment, right? So very strong agent is radiation, right? Why radiation? Because the particles that emitted by X-rays, right, they actually go into the DNA molecule and interfere with the DNA replication. Okay. What chemicals can cause mutations? Yes, tell me. No, talk to me.
The chemicals?
Mm-hmm.
Chemicals or animals? Oh.
Smoking can cause mutation, as we see in the picture. Consumption of radiation can cause mutation. Consumption of bad foods. Well, everything around us is technically radiation. I'm sorry, everything around, no. Mutation can also be natural. Mutation can also be natural, such as something happened as you grow up. Maybe
I should say very
you take
like
a sip
natural
of that.
radiation is sun radiation. Sun radiation can cause mutation in your skin cells, and that can cause what? What these mutations can cause? Hmm?
Is it cancer?
Absolutely, they can cause cancer. Actually, cancer is considered, like aging disease because during lifetime, people accumulate mutations. Mutations and mutations and mutations, and these mutations eventually can cause wrong formation of the cells, which is cancer. And that's what I said. Basically, genetic diseases pretty much could be caused by these mutations, right? So mistaken the DNA, non-functioning enzyme, certain chemical reactions could be carried out, and this can cause genetic disease. And this is it. So this is it about chapter 6, right? And I promised you that one more, but very, very brief. and very brief is biotechnology. Okay, so biotechnology is actually a scientific approach to use knowledge in molecular biology, in genetics, to for our own, as it actually said here, practical benefits. Okay, so genetic engineering is actually manipulating genetic code on the organism deliberately, right? Moving genes from here to there, right? And that could be used in agriculture, right? And it's done actually for years, for very long years. It could be done for health benefits, and it's also can be done in forensic science, right? All this knowledge. Okay. Now, guys, if I go back to whiteboard and I go back to viruses, right? So virus, remember, is a piece of DNA and RNA enclosed in the protein shell, right? And remember I explained to you that virus can get inside the cell and can insert its own genetic information to your genetic material. So in this sense, virus is called vector. Why vector? You take viral genetic material, you take a piece of it, right? You cut the bad part, like disease causing, you cut it, and you add new gene that you're interested in, right? So this virus is going to get inside the cell and add the genes that actually can be beneficial. So this is one of the ways how to treat genetic diseases, right? So genetic disease could be actually treated by inserting functioning genes into the cells, which is kind of like a very, very general approach, right? Because there are many cells, right? And you can't guarantee that every single cell will be targeted, right? But this is the attitude, that this is the approach. So that's a reason why I explained to you about the viruses at the very beginning, because viruses can carry genes, right? Viruses could be modified and they could be used for our benefits. Okay, this one, for example, another application. Let's say this is the whole plasmid and this is CRISPR. Okay, I'm gonna talk a little bit, just a little bit of every single thing. Let's say you're all familiar with the disease called diabetes, yes? What is diabetes?
Which type?
Doesn't matter which type. Let's say type 1.
Well, isn't diabetes when your pancreas can't form insulin?
Excellent. Excellent. Yes. So diabetes patients, they depend on insulin. So insulin was, again, the molecular structure of insulin was discovered beginning of the 20th century, which is a huge breakthrough. It basically saved so many lives, people who suffered from diabetes for years and they didn't have anything available to make their lives better, okay? And all the people were dying from diabetes as well. So now, the molecule was discovered. And it's not an easy thing actually to produce this molecule in factories in commercial amounts, right? And nowadays, insulin is not made on the factories. Probably it is, but and nowadays, gene of insulin is isolated, amplified, inserted into bacteria cells or viruses. And actually bacteria produce insulin for you. Bacteria cell have ribosomes, so they can make any protein. Just feed them with genetic material. So this is actually a core of biotechnology. You use bacteria, for example, you use other living organisms, simple living organisms, as a factories of, factories that can actually synthesize molecules that you actually need. And they can do it so much better than, for example, chemists in the labs, okay? Antibiotics produced this way. So many medications actually produced this way by biotechnology. And I think not only medications, I think there are so many other substances that actually used in medicine made through biotechnology. Something that I want to show you, this is just like details. Plasmid is a piece of DNA that carries genes that could be inserted into bacteria, right? From bacteria to bacteria. Again, genes could be inserted through the viruses. And that's how, again, bacteria can produce desirable molecules. I don't know if you've heard about CRISPR. CRISPR is actually a way to edit DNA. It's an enzyme that can kind of like cut a piece of DNA. I don't know if you heard about, I think last year, LAP. That was a story in China when there was a doctor who genetically modified twins, right?
Yeah, he was arrested.
Yes, he lost his license, right? But he actually genetically modified 2 embryos and made them resistant to HIV. Okay, which is actually a beneficial thing, right? Being resistant to HIV, but he had no right to do that.
If I'm allowed to pontificate a little bit, he took 2 embryo cells, which are very small. You can put them in a Petri dish, and inserted them with some sort of I don't know what he inserted them with, some sort of thing that made them that would make them genetically predisposed against
HIV. But he was yes.
And then he would take the 2 embryos that he embryo cells that he did them with, and then showed it to the camera at the SCMP. I remember that day quite well. I saw it in the newspaper online. He was showing the Petri dish to the camera. That got him in trouble. And for some reason now we don't see or hear of them anymore. It wasn't actually anything.
Yes.
What?
Thank you so much. Yes, exactly. But this is the tool that he used. He used the CRISPR that could be used again for very beneficial things, right? But I don't think that actually it's ethical to genetically modify embryos, even for doing like making them resistant to HIV, right? But CRISPR, again, it's actually a hope for people who have genetic diseases, right? This is their hope because CRISPR can actually help remove faulty genes, and there's a way actually how to insert the right genes. So you see potential of CRISPR is mosquito DNA can be altered to block malaria from spreading. I think in Brazil, when there was a Zika outbreak, have you heard about Zika, guys?
Yep.
Zika virus.
Yeah.
That was like a couple years ago.
Yes. they were doing this actually. They used they altered mosquitoes' DNA actually to prevent mosquitoes from reproduction, which helped them to stop their epidemic. Okay, this can be used actually to alter white blood cells in humans and target them against cancer, and that could be used also like. The eagles have been engineered to be born with twice as muscle as a typical dog. So. But these risks, okay. So the last thing that I want to talk about. So I really want you to read this chapter, but there's something that I'd like to talk to you right now. Okay. So. Do you remember the movie called Jurassic Park?
Yeah.
Yeah. So do you remember the story how they made a dino?
I don't remember.
They found the fossil of a mosquito from that period with some sort of genetic material. Then I don't know if it was blood or anything. And then they extracted it, found all of this dinosaur DNA, and then started growing it in labs.
Exactly. So they found a fossil mosquito. In mosquito's stomach, they found blood from dino, right? So from that blood, they actually kind of like.
Which is kind of amazing. I don't think blood would still be liquid or in that form. I don't think. I mean, I don't know. It just.
This is a fiction, of course. This is a science fiction. Okay, so they got dino genome. Then they took step 4. They took emu or ostrich. Ostrich egg, right? So they took an ostrich egg. Then the ostrich egg, remember, there is a nucleus. So took out ostrich eggs and put dino genes. And then magic, a dino hatched. And it looks more like a squirrel. Dinos don't have ears like that. Like then dino hatched from the egg, right? This is, again, it's not scientifically possible because first of all, yeah, it's DNA being intact inside their mosquitoes cells, like inside mosquito is definitely, after millions of years, not really possible, right? Even if you can extract some DNA, you can extract DNA for the whole entire dino. It's really, really not possible. Okay, but the whole idea could be applied to animals that are kind of like existed recently. So animals that existed recently. Right? So what do you think? When was the last time mammoths have been seen around? How many years ago?
Can you repeat that, Professor?
Just 1 second, guys. Just 1 second, 1 second, 1 second. Вася, дай мне. Yes, the question, how long ago dinos have been seen?
Priviyati. Maybe a lot of, definitely like millions and millions of years ago.
This is my sister.
So cute. Is it he or she?
This is a boy.
The boy.
Yeah. And I think he hates me. I gave him a bath.
Cute. Oh, you gave him a rat. Yeah, maybe that's why.
Yeah, I think he's like, he holds grudges against me.
We're talking
Okay.
about a dog.
I
What?
got you right now, Professor. Look.
So how long ago last time mammoths been around? Not millions of years.
Like,
400,000?
Believe it or not, 3,000 years. Right?
Oh.
Yes, and some of them actually found frozen and well preserved, very well preserved. So you can actually get a hold on their DNA, like good DNA. So next thing, what you can do, you can take the mammoth's DNA, right? Take the egg cell from their close relative, which is called, who's the close relative? Come on. Who's close relative of mammoth?
The elephants?
Elephants, yes, you take elephant. You take elephant egg cell, right? Pull out DNA of elephant. Insert DNA of mammoth. And here you go. An elephant gonna give birth to mammoth. The question is, what are you gonna do with this mammoth? What do you think?
I'm back, Professor.
I see it. What are you gonna do with the mammoth?
Look at him, he's so fat.
Wow, that's a big one. Can you show him the camera a bit closer if he's not gonna resist?
We don't get along though. I get along with my other one.
You have two cats?
Mr.
Yeah. You see, he's mad fat.
Put it closer to the camera.
Garfield.
Is that actually his name?
No, his name is Lucho. Typical Hispanic.
Adorable.
My cat has typical Russian name.
Is it
Vasia.
Privyat? Is what? Oh yeah, I'm about to feed them, so I'm gonna turn my camera off.
Okay, so we're talking about how you can bring back mammoths to life. So again, the question, what are you gonna bring it for? You're gonna bring it for the sake of science, but this mammoth's gonna be alive. So they're gonna exhibit it in some parks. You know, it's like there are a lot of things that you should take in consideration. Just, okay, it's possible. And yeah, so far, it's still a project. I read like a big article on this about mammoths, but there were several attempts actually to bring back animals who went extinct recently. So I think there's some like mountain bighorn sheep from Spain. They did similar manipulation. So they took this DNA from their, again, extinct, put it to egg cell of very, very close relative of that sheep that is still alive. It was like partially successful. So they were able to get some of them born, but unfortunately they didn't survive for too long. So I think there's like trials and errors. But again, molecular biology can be a tool to help to preserve certain species that unfortunately so many of them go extinct right in front of our eyes. Can you name animals that went extinct recently?
The albino gorilla. The albino gorilla.
Yes.
Aren't polar bears about to go to extinct?
Unfortunately, no. I think they're endangered for sure, but
Oh, the rhino. Some type of rhino.
type of rhino.
Yeah.
Absolutely. I think like there was like Tasmanian wolf, very, very famous animal that went extinct. Dodo birds, right?
Are we talking about old animals?
Yes, I mean those who like for the past like 100+ years went extinct. There were like interesting pigeons in the United States. They were swarming in huge amounts over the country. They called passenger pigeons. Oh, look at him.
Yeah, I know. He's so bad.
Passenger
You
pigeons.
should be a cat by Professor.
Yes, I see. And these pigeons, people really used to shoot them. It was like for fun. So they were shooting and shooting them. And even though they were in really great numbers, they went extinct 100 years ago. Okay, you got the point how biotechnology could be used, right? And the last thing, I don't know if you heard about cloning.
Yes. Wasn't the sheep's the first thing to be cloned?
Yes, she, her name was Dolly, right?
Dolly, yeah.
What do you basically do? You can collect any cell, right? Any cell from the animal. You collect their DNA from the nucleus, and then you use the egg cell of another animal, take the nucleus from the egg cell, and insert this DNA in, right? So this is cloning. Some people who, I believe, who have so much money that they don't know what to do with them, what they do, their favorite pet. I'm not sure about cats, but I know about dogs. So yes, your pet dog dies. This is really heartbreaking. And what they do, they pay money to get this dog cloned, right?
You can actually pay for clones now?
I think it costs like only $30,000.
Really? I didn't know that.
Yes, $30,000. And I know there are companies in China. I know there are companies in South Korea who do that. So basically you send them the specimen, right? And then they actually ship you a clone of your pet.
That's pretty crazy. Yeah.
Mm-hmm. Yeah. So this is again just like how like biotechnology, how this knowledge of genes and proteins can be applied. But I think it should do more than just to entertain people who fancy cloning their favorite pets. Okay, any questions? So listen to me. Quiz will be on chapter 6. Chapter 7, I only want you to read, okay? Now, if you see assignment for chapter 5 on Launchpad, ignore. You don't have to do chapter 5 assignments. I didn't cover it, okay? You can do assignments for chapter 6 and 7, but quiz will be only on chapter 6. I'm also gonna post it. So let me just take an attendance and we'll be done for today.